Hello everyone, please answer your bell work that is posted right here. Go ahead and pause your screen before we get into the lecture on proteins for today. Okay, so topic 5 is our third macromolecule, proteins, and proteins are the most abundant organic molecules and they have a diverse range of functions. They can help regulate body functions. They can have a component and structure to the body. They have protective functions, transport functions. They can function as enzymes, like we spoke about earlier, which helps speed up chemical reactions. And proteins can also act as toxins. And here again are types of proteins listed. So digestive enzymes, for example, amylase is going to be the enzyme that helps break down carbohydrates. Lipase is the digestive enzyme that's going to help break down lipids. Pepsin is the digestive enzyme that, as a matter of fact, helps break down proteins, and trypsin is another enzyme. For transport, one example of a transport protein is hemoglobin or albumin. Hemoglobin and albumin help transport substances in the blood or lymph throughout the body. And one way of thinking of how do we transport things through the body? Well, most of the things in our body are going to require liquid to be transported from one area to the next. And a big component in blood is plasma, which is composed of liquid. And lymph is also composed of lymph, which is a fluid. Hemoglobin acts to carry substances in the blood, in particular oxygen. Hemoglobin is our oxygen delivery system, which is a very important function to get oxygen to all of the tissues in our body to prevent hypoxia. In structure, we can think of actin, tuberculin, or keratin, and these act as a structural function in proteins, and it helps construct different structures like the cytoskeleton in animals. Keratin, in fact, helps us build our hair and our nails for different types of structures in our body. Another example of proteins are hormones such as insulin and thyroxine, and these hormones act as chemical messengers that are able to coordinate activity of different body systems and to communicate with different parts of the body. This is another example of, a need for the fluid in our body. So insulin uses the blood to help deliver glucose to the tissues, for example. And so does thyroxine. Thyroxine is going to travel throughout the blood to coordinate with different body systems to help keep that homeostasis throughout our body. Proteins can also act as defense, like immunoglobulins, which help protect the body from foreign pathogens. We have contractile function of proteins such as actin and myosin, which are both important for muscle contraction. And proteins can also act as storage, such as legume storage proteins or egg whites, and those provide nourishment and early development of the embryo and the seedling. I know we think of legumes and eggs as things that we eat, but eggs are also used in reproduction. And one function of the egg in, for example, human reproduction is to continually feed the embryo with the necessary nutrition that it needs. Now, I wouldn't say you need to memorize each of these examples, but knowing a couple off the top of your head will help you understand these different functions of proteins. I would say go ahead and memorize that 1, 2, 3, 4, 5, 6, 7 these 7 are all functions of proteins. Okay, so we talked about proteins as enzymes. Enzymes are catalysts, and catalysts speed up chemical reactions. So for example, when we talked about the dehydration synthesis reaction, an enzyme can be at that binding site of those different molecules and enable that to have an easier binding and lower the necessary energy needed to complete that chemical reaction. We can have catabolic enzymes, which break down substrates, such as those that we use for digestive enzymes. And we can also have anabolic enzymes that are going to help speed up the building of more complex molecules. So for our dehydration synthesis reaction, that would be more of an anabolic enzyme that we would need, because for dehydration synthesis, we are trying to build a more complex molecule, right? So we take these monomers from our macromolecules, we add an anabolic enzyme, and then we are able to build a more complex substance in a faster manner. And the opposite is for catabolic or catabolism reactions or catabolic enzymes, where, for example, we have lipase, which is going to help our body break down carbohydrates in a faster mechanism. So we can take a disaccharide or a polysaccharide and add lipase, the catabolic enzyme, to help break it down into those monomers in a quicker way. So those are two examples of catabolic and anabolic enzymes. Now, enzymes are specific to the chemicals that they're acting on. An enzyme for lipase, for example, that catabolic enzyme we just spoke about, is no good for proteins. Lipase is not going to help you break down proteins. We have a different type of enzyme for that. And the same is true with anabolic enzymes. They are specific only to the chemicals they are acting on. The monomer of proteins are amino acids. So amino acids bind together to form these different types of proteins. There is a fundamental structure which you need to memorize for amino acids. The fundamental structure of amino acids is a central carbon atom, an amino group, which is NH2, a carboxyl group, which is COOH. Remember, we also saw that in the lipids, so don't get these two confused. A hydrogen, and a side group R chain. A lot of people get confused on what we mean by the side group R chain. That means a chain of different amounts of carbons in a row. That's pretty much all an R group means. It means that there are different numbers of carbon that can be chained together to form an R group. And here we can see the model of what this amino acid would look like. So we have the amino group, which is the NH2. And here in the middle, we have the central carbon bonded to another hydrogen. And that carbon will also be bound to the side group or the R group, which has a various ability to have different numbers of carbons. We will see more specific examples on the next page. And then we have the carboxyl group, where we have a carbon double bonded to oxygen, and that carbon is also bound to an OH as well. Let's look a little bit more into our different amino acids. Oops. Here we have listed the 20 different amino acids. Now you can probably do a quick Google search to see the different structures of these different amino acids, but you will notice that they follow that same pattern that we just spoke about, where there is an amino group, a carbon, central carbon, bound to a hydrogen and an R group. This R group is what varies on the amino acids. And then we have a carboxyl group. So when you look at these pictures of amino acids, I understand that this slide might be a little hard to read because it's small, but they all differ in only their R group side chain. That is the only difference between these 20 amino acids. These 20 different amino acids are able to bind in different patterns and different ways to form our proteins throughout our body. And if you notice, like I just stated, the fundamental structure is the same, only the R group changes. Now, you don't need to memorize these different amino acids, but we will probably touch on amino acids throughout the rest of the year. How do these amino acids bind together to form our polymers, right? We said that amino acids are the monomers of proteins, and these monomers have to link together to build the polymer of a protein. These monomers are linked via peptide bond formation, and a lot of times you will see this called polypeptide bonds. And polypeptides mean there's many peptide bonds. And this happens through a dehydration synthesis reaction, which we've already touched on throughout this chapter. As you can see, the two carbon chains right here, react through their carboxyl groups, and they go through the dehydration synthesis reaction where water is lost. And that is how our peptide bond is formed. Now you'll notice, unlike the glycosidic bond that links carbohydrates together, this peptide bond is linked through a nitrogen, which is interesting. That's why it's a different bond type than the glycosidic bond, because we have a different combination that this dehydration synthesis reaction results in. We still lose water, but the bond is a different type of bond. Okay, so now we're going to get into protein structure. Protein is based on 4 different levels of structure: primary, secondary, tertiary, and quaternary. A protein's shape is critical to its function throughout the body. So we're going to get into different types of structures. So first we're going to talk about primary structure, and it is the unique sequence of amino acids in a polypeptide. And this is based on DNA from encoded genes. So we get gene transcription, translation, and that is typically forming proteins. So this is based on the genetic code, and we will get into more about that later. But what you need to understand is primary structure deals with the sequence of amino acids. One example of a mutation in this protein structure that results in a disorder is sickle cell anemia. What happens is, in our amino acid chain, we are supposed to have a glutamate, and instead our genes told us that a valine goes there. And these are 2 different amino acids. And while it seems that one little change in one amino acid, where there is such a long chain of amino acids, wouldn't be that detrimental, but it does. It results in a change in the primary structure of the protein hemoglobin. Instead of having that round biconcave shape of a red blood cell, it results in the sickling of the red blood cell. And this differentiated shape of the red blood cell results in many problems for the patient, including occlusion of vital organs or of blood vessels. It has a lower carrying capacity of oxygen, so hemoglobin is not as effective in carrying oxygen to deliver them to the cells, which is one of the important functions of proteins of transport. And it's also very painful to the patient. Here is a picture of our red blood cell smear from a person with sickle cell disease, and you can see those little hook-like, skinny shapes are the sickled cells, whereas the red blood cell is typically round. Our secondary structure deals with the local folding of the polypeptide. And there are multiple ways in which we can get this secondary structure. One is due to the hydrogen, there's one way that we get this local folding structure, and that's between our carboxyl group and amine groups, and 2 protein chains, or 2 amino acids chains lined next to each other. When we have hydrogens in similar vicinity to each other, it allows for cross-bonding between hydrogens, forming this pleated and helical shape. So if you see a helical or pleated shape, know that that is the secondary protein structure, and it is due to hydrogen bonding. Those are 2 important facts of this slide. Tertiary protein structure is the unique 3-dimensional structure of a protein, and this is formed via chemical interactions between R groups and amino groups. So we can have various chemical interactions between these R groups and amino groups within the proteins to form these bonds. And here are the 4 different types of interactions that occur at the tertiary protein structure. One is hydrophobic interactions. So if one part of the R chain is hydrophilic and one part is hydrophobic, well, those are not going to be compatible, and they will certainly not be as attracted to each other, or sometimes they are both hydrophobic and they're both more attracted to each other. So that is one example of how tertiary proteins folding will result in different shapes of our protein. Ionic bonding is another one. So when we have 2 chemicals with similar or different charges, and they're more attracted to each other, for example, positive is more attracted to negative, then these two will want to be more closely related. Hydrogen bonding, like we talked about in the previous slide of secondary structure, is another option for tertiary protein structure. So it still has the ability to have hydrogen bonding across the amino acid groups to form this tertiary structure. And the last one is disulfide linkages. If you were to go back and look at some of these R groups, some of them have sulfur on their as part of their group. So you can have disulfide linkages between the sulfurs of two different amino acids, which is also called a disulfide bridge. Sometimes you'll see it called disulfide bond, but right here we call it a disulfide linkage. So that's another important part to know, that disulfide linkages result in tertiary protein structure. And finally, we talk about our quaternary protein structure. And this occurs because we have interactions between several polypeptides that make up a protein. So we have multiple different strings of polypeptides that have interactions between each other. And these are going to help stabilize the final quaternary structure of proteins. Here is another picture from your book at the 4 levels of protein structure. So you can see primary structure is the chain of amino acids. Secondary structure is either the helix or the pleated sheet. And then we get the tertiary structure, where we have 3-dimensional look at our proteins. And if you notice, the secondary structure was 2-dimensional. The primary structure is 1-dimensional. And then the quaternary structure is the overall finalized and most stable structure. Okay, denaturation of proteins is still part of proteins, and it is our other topic. Denaturation is changes in protein structure that lead to changes in function. So, for example, heating an egg to an extreme temperature can result in irreversible denaturation of the egg protein. So we go from a liquid to a solid. So you can boil an egg, and you can see that protein becomes from a liquid to a solid. Chemical interactions are broken through changes in pH, changes in temperature, or interaction with different chemicals. So you can even denature proteins sometimes by having them in the microwave. It goes through the microwave, it changes temperature, and it causes a variation in the protein structure. And this will conclude our lecture. Please stay tuned for our exit ticket for the day. And here's your exit ticket for the day. Go ahead and pause your screen. Let me know if you have any questions, and I look forward to seeing you all soon. Have a great rest of your day.